We're going to look today at mushrooms, and we'll follow this theme of way cool. Some parts of this talk are drawn from a book I just published a few months ago with Oregon State University Press. I brought some copies along if anybody's interested. It's called Boundary Layer. The book is a reflection on boundary layers in nature and also boundary layers in concepts about nature. I'm kind of a conceptual guy. Boundary layers, I don't know if you remember this from, maybe this was the physics class you slept through, but boundary layers, it's a term from physics, and boundary layers emerge when you have two large stable systems flowing against each other. So if you have a solid and a liquid like air, when they flow against each other, it creates this special boundary layer. And we learned long ago, early part of the 20th century, that these boundary layers have very specific characteristics. And they aren't just blends of the two systems around them. They create their own rules. And they're creative blends of what's in the bounding system. So I took this metaphor, and I'm not to ever grab this metaphor. It's been used before. We have atmospheric boundary layers, oceanic boundary layers. I just took it a little further, and I'm trying to apply this term to what we find on the floor of forests here in the Pacific Northwest. I realized, I came here 12 years ago from Ontario, and I realized when I came out here, I was an amateur naturalist in Ontario. When I came out here, I continued my work, and I realized that there was something very special going on these forest floors. And it was very much like a boundary layer. And it had in it mosses, fungi, lichens, small vascular plants. So a lichenologist and I, his name's Trevor Goward, he and I, yeah, you know Trevor, right? So he and I wanted to come up with a term for this. So we came up with the term stegnon. Okay, we invented it. Right, so don't go looking in the dictionary for it, but yet. But it's on an analogy with plankton. Just as the plankton in the ocean really form the basis of life in the ocean, and yet the plankton are not monophyletic. They're all kinds of different creatures. They're plants and animals, right? And yet they are the basis of life in the ocean. So on the floor of the Pacific Northwest forest, this is the basis of life in these forests. It's the stegnon. And then I go on and deal with some conceptual boundary layers that arise from this, having to do with species and ecosystems and the term wilderness. I also, in the book, it's the first time I write books, and usually I'm the speaker in the books, right? And I'm talking about something else. But in this book, I tried something different. I tried to bring in other voices. And so I have in this book 10 innovative scientists who are studying these groups of organisms. I found some very interesting people. We got a couple of them sitting here tonight. And all I can say is that mushroom people can be a bit weird. But believe me, they can't hold a candle to bryologists, to people who study mosses. These people are, wow, okay? And it's very interesting people, right? And so they were kind, they were generous, they opened up their lives to me. It's not exactly a kiss and tell book, but they allowed me to put things in print that had never been in print. And I think you'll find that engaging in the book if you get around to reading it. In the mushroom chapter, I discuss the work of Adolph and Aluna Cheska. That's Aluna over there. I just spent 4 days with she and Adolph at what we call the Pacific Northwest Key Council. It's a group of scientists who get together and work on classifying mushrooms. As I say, the aspect of the stegnon that we'll deal with today are the fungi. So when I moved to BC, I was quite surprised. I know there are fungi in Ontario, and there's a great diversity, but at least where I was, they weren't very prominent. I was quite surprised when I came out here to discover the second spring. You know, spring, the wildflowers come up, you get the colors and the smells and all these wonderful things about spring. And here, we get the same thing in the fall, only it's mushrooms, not plants. You get the colors and the smells. The only problem is spring is followed by the nice summer. The second spring is followed by winter. But other than that, it really is a kind of second spring. They bloom, that's a plant word, but that's why I put it in quotes, in a wide variety of shapes, colors, and smells. So we'll explore this second spring of mushroom in 3 phases, and in the course of it, we'll inflect this adjective cool. I know cool is not cool anymore, right? But you know what I mean by the word. So first of all, I want to look at an aspect of that I'm really interested in fungi, and that's how one fungi makes another. And we'll look at the science of that. So that's cool. Then we'll look at something even cooler. I want to look at mycorrhizal mushrooms, another really hot topic in mycology right now. And then finally, we're just going to look at some different kinds of mushrooms. And I think you'll find this the coolest part of the talk. So first of all, a little bit about fungal science and sex. Mushrooms, of course, are fungi. Mushroom caps are the fruiting body. Again, put it in quotes because that's a plant word, fruiting, right? They're the fruiting body of certain kinds of fungi. The real fungus, the real mushroom, if you like, isn't that cap you see. It's out there in the soil. It's an almost invisible network of very small threads we call hyphae, and together they make up a mycelium. And that's what lives from year to year. The cap is simply like a berry on a bush, sent up to reproduce. Some years they send up caps, some years they don't, right? Depending on the mushroom. Interestingly, fungi are not plants. In the first half of the 20th century, though, if you would have gone to university before 1950 and taken a botany course, in that botany course, you'd have probably spent a week on mushrooms. They were actually covered in botany courses, even though back then they knew they weren't plants. But it's become very clear since about 1950, we've moved them out of botany courses. And we're treating them now as what they are, their own kingdom, right? Kingdoms. You know that term. That term kingdom. In terms of the now dated system, now we recognize 9, 11 kingdoms. But when we first really became aware of this in the 1950s, we had the 6-kingdom system of Carl Woese. And he listed in the kingdoms the bacteria in one kingdom, and he put the strange bacteria in another, and then the one-celled animals and the algae, the protista. But here are the big 3, fungi, plants, and animals. What you really want to note out of this is that fungi are in their own kingdom, right? They're not plants, and they're not animals. Interestingly, from an evolutionary point of view, fungi may be closer to animals than to plants. For one thing, both animals and fungi are heterotrophic, and plants are autotrophic. What we mean by that is plants go out and absorb sunlight and create their own carbon compounds out of carbon dioxide and other chemicals, right? So they can live on their own. They don't depend on any other kingdom. They're autotrophic. But the heterotrophic kingdoms eventually have to consume what the plants make. And we're heterotrophic. You eat plants. I know some of you like to feed them to animals first, but ultimately you are eating plants, right? And fungi are the same. They need this created material in order to live. Fungi, even if they're green fungi, they don't photosynthesize. No more than a young university student who dyes her hair green is photosynthesizing when she walks across campus. Also, fungal cell walls contain chitin, which is an animal protein. If you remember this from high school, you know, the exoskeletons of ants made out of chitin. And so are the cell walls of most fungi. They're not made out of the lignin and cellulose that the animal cells are made out of. And then, since we've been able to do genetic analysis, we've also begun to understand that they're more closely related to the animal kingdom. In fact, here's one of these tree of life diagrams. You can find any number of these. Everybody's trying to figure out where it all got started and how everything is related. But up here in the upper left, you'll see, in the upper left here, you'll see the plants, right? They've colored it green handily. So let's go looking around and see where the animals are. Whoa, we go down here. There they are, right there. What's that right above them? Mycetes. Ah, those are mushrooms, right? So, look, there's the branch. You see? It happens much later. And in fact, in this system, which is based on the work of the Oxford evolutionary biologist Thomas Cavalier Smith, he calls all of these things uniconts. So fungi and humans, animals, are uniconts. So you see we're more closely related than the plants are. So a bit about where fungi came from. They first appeared on the Earth 1 billion years ago, we think. They were, of course, water-based until about 500 million years ago when things began to emerge on the land. And we think they may have come out on the land with the very first plants, which would be something like mosses, right? Very primitive plants. We can break this kingdom of fungi down into separate parts, subkingdoms, if you like. We call these things phyla now. And currently, about 7 or 8 different phyla are accepted by scientists within the kingdom fungi. We're only concerned today with maybe 3 of them. We're going to talk about the mushrooms you see out in the woods, and they're all Ascomycota and Basidiomycota. But we'll also talk for a moment when we do mycorrhizal mushrooms about another phyla, which is the Glomeromycota. And you may have heard lately about a lot about chytrids because of the amphibian die-off, the frogs and salamanders that are being killed by this fungus. But that's really the part of the animal kingdom we're interested in today. Researchers estimate that there may be 1.5 million different fungi out there. This was computed by a man named David Hawksworth in 1990, and he updated his work in 2001. The way he came up with this number is a ratio. We find when we go out and do studies in the field that generally we can find 6 different kinds of fungi for every vascular plant. So since there's 250,000 vascular plants, then we guess there are 1.5 million fungi. Most people today think that vastly underestimates the number. And even Hawksworth says if you start counting cryptic fungi, we could have another 5 million. So there's a huge number of fungi out there. And so how many of these do we really know anything about? Well, there's only about 100,000 names for fungi in the official lists. So you can see that we know less than 10% of them, probably more like 2 or 3% of them. Of course, when it comes to ones that produce mushrooms, because we see those more obviously, we know more about those. So I would guess maybe you could guess that we may know 20% of all the mushroom-producing fungi in the world. It's a vast number out there that we know nothing about. Even in BC, this weekend I was with a bunch of taxonomists, and one of the things we're doing is looking for new species. And gosh, there were several there that may end up papers written about them and going down as new species before we're done. They just didn't fit anything else, right? And we found some that were unknown in BC before. So let's take a quick look at the mushroom life cycle. I find when you're studying various kinds of groups of organisms, if you really want to get a handle on what's going on, you should look at the life cycle. Find out how one organism gives rise to the next generation of organisms. And the same thing is true of fungi. If you really understand this, you begin to understand how peculiar and cool fungi really are. Generally, when you show a life cycle diagram, it's divided into 2 parts. One part will be what we call haploid and the other diploid. Yeah, maybe you slept through that class too, right? Haploid and diploid. No, some of you know what haploid and diploid are. We are diploid organisms. We have 23 paired chromosomes in almost every cell of our body, right? However, once in a while, we become haploid, and that's when we want to make a new generation. So sperm and eggs are haploid, right? For a brief time, then they come together, and bang, we've got a diploid organism again. So we apparently do not care much about being haploid. We want to be diploid. Other things like mosses spend most of their life haploid, and they just get diploid long enough to get to the next generation. But you get this division, haploid and diploid. We see immediately a problem here. We got 3 divisions here, okay? And it's this one over here that's peculiar. We have haploid, diploid, and dikaryotic. And what we call a mushroom is dikaryotic. It's not haploid or diploid. which immediately should raise some flags. You say, whoa, what are we dealing with here? This is not an animal. This is not a plant. This is something very different. Let's look at what that dikaryotic means. We're going to follow a mushroom through its life cycle. Mushrooms begin their life as haploid spores with only half the number of genes, just like sperm and egg for us. And these are a picture of Agaricus bisporus spores. Do you know Agaricus bisporus? Sure you do. It's the button mushroom you buy in the grocery store, right? And these things are spores, right? Here and here and here. This is not a spore. This is a spore-bearing body called a basidium. And you can see the spores are starting to mature on the basidia around here. Right? So we start off with spore. It's very small, 5 to 10 microns. You cannot see it with your naked eye. When that spore lights on a suitable medium, it will germinate. It will, it's a plant word again, but it will begin to grow. So it sends out a thread. You see it here. We call that a hypha. All of them are hyphae, and when it gets complex enough, we call it a mycelium, and it starts branching. And they go out in the soil, and they form these hyphal nets, okay? And it grows. Now, at some point after the mushroom is grown, it starts to think about baby mushrooms. How do I get to the next generation? So it goes looking for a compatible partner. And when it finds a compatible partner, I don't want to call them sexes, because that would imply there's a male and a female. Well, we don't have males and females here. All we have are homokaryons, compatible partners. We know one mushroom, for example, that has 42 different types. Could you imagine a mushroom singles bar? Yeah, everybody wearing name tags, and you spend all evening going around trying to find a compatible type, right? But when they find compatible types out there in the soil, their hyphae fuse. They build a bridge between them. And when the hyphae fuse, they're getting ready to go to the next generation. Here's one hyphae, one mushroom. It has the pink colored. These are nuclei inside the cells. Here's green nuclei. And when they fuse, the cell walls start to break down, and they build these little bridges from one cell to another, we call clamps. And the nuclei start duplicating, and they flow through both mushrooms. So pretty soon, we've got one big mushroom with 2 nuclei in every cell. Cell walls reform. Now, this is where, if you come at this from an animal point of view, you'd expect the nuclei to fuse, and we'd get a new mushroom, but they don't. They stay separate. So these cells are dikaryotic. They have 2 nuclei in them. And the mushroom stays that way for a period of time until it's ready to reproduce. And when it's ready to reproduce, it forms a little knot in the mycelium. This is a hyphal knot. This is a primordia, we call it. This is a primordia of Psilocybe cubensis. That's the one you order by mail, the magic mushroom. So it forms this little primordia, and then the primordia elongate with a stipe, and a cap forms at the top in a gilled mushroom, and gills form on the cap, and under the cap there's a little a veil that keeps the moisture in, and until it's ready to drop the spores. And when it's ready to drop the spores, so here we have the mushroom, right, with the gills. If you did a cross-section of the gills, it'd look like this. And all along the walls of the gill are these basidia, the spore-bearing bodies. The basidia form here, and the spores form on the basidia. But remember, this thing is dikaryotic. So the basidium starts off as dikaryotic with 2 nuclei, and when it's ready to put those spores out, finally, after all this time, the nuclei fuse. And here you have the new mushroom with a new genetic signature. It goes to cross over all the other neat things that animals and plants, nuclei do when they fuse. And once it's fused, then it undergoes meiosis and mitosis and produces 4 haploid spores. So here we are, ready to go out to the next generation. So that's the life cycle. See what I mean? We've gone from spore to spore now. And you can see just how cool and peculiar mushrooms really are. Here's a, you might be interested in this. When these spores form on the basidia, when they're mature, the basidium drops the spores. It actually has a way, using water pressure, it can plunk them out a little bit, because what it wants to do is to get them into this space, and this is a cross section, here's a gill. It wants to get them into this space between the gills so they can fall to the ground. And in a typical mushroom, it takes about 3 minutes for a spore to hit the ground, if it ever gets there, because it's very light. The winds can carry them everywhere. And in some mushrooms, which I'll talk about in just a moment, the ascomycetes, the spores are actually shot out. Down at the bottom here, you see a little film. This is a morel ascus discharging its spores. You see that again? There we go. Zoom. There are 8 of them, and I'll explain why in a moment. But it actually shoots them out. And in ascomycetes, such as cup fungi, you can see this. If you go out and find a cup fungus in the woods and you blow on it and warm it up, all the asci can shoot all at once. And you normally can't see spores, but all of a sudden you get this huge cloud of spores. And it's so thick you can see it. And they are violently discharged. But with the basidia, they just sort of pop off. Okay, so a typical agaricus type mushroom usually puts out spores for up to a week, at about 1 billion spores per day. An English study found that during mushroom season at head height, there are about 10,000 spores per cubic meter. So over the next month, you are going to breathe in a lot of mushroom spores. They will not colonize your lungs under normal circumstances because you have something called an immune system. But if you haven't got an immune system, and there are people who don't, this is a problem, right? Mushrooms can actually colonize people. For producing spores, the champion spore producer, and it's on this cart that's up above there, is Ganoderma applanatum, the artist conch. It can live for several months and produce spores, and over its lifetime, in a year, it can produce 5 trillion spores. End to end, those spores would reach around the earth. So the life cycle I've just described for you is that for a gilled mushroom, it's a basidiomycete, and it's a mushroom-producing fungus, produces macrofungi. There are also these ones we call ascomycetes, like morels and cup fungi and some things like this, and they have a very similar life cycle to what I've just covered, except instead of holding the spores in basidia, they hold them in these sacs we call asci, and as I showed you, they can shoot them out rather quickly. However, many fungi, as far as we know, don't do sexual reproduction. They just clone themselves, right? And they seem to get along perfectly well like that. And in addition, of the fungi that do sexual reproduction, perhaps 1/3 of them have a completely different life cycle than what I've described here. The molds and the mildew. So if you want to find out about this, I recommend Bryce Kendrick. He's a member of our Mushroom Society, a world-renowned figure on these cryptic fungi. His book, The Fifth Kingdom, I think it's 3rd or 4th edition now. But it's a cool book, and Bryce is so cool he's cold. Okay, so that's the cool part of the talk, right? Mushroom science and sex. Now, let's look at the cooler part. I'd like to talk a bit about one of my favorite topics, which is mycorrhizal mushrooms, because that has a lot to do with what you're going to see in the woods over the next month. We could start off thinking about this by thinking about how mushrooms make a living, right? Except for lichens and lichenized mushrooms, mushrooms have no chlorophyll, as I said. Only plants do. So where do mushrooms get their energy? And it turns out there are really 4 places mushrooms can get their energy. So a few of them are actually carnivores. For example, Lacaria bicolor. When springtails feed on it, there's a poison in it that kills them, and the springtails drop to the ground, and the mycelium absorbs their nitrogen. It's actually eating the bugs. There are some mushrooms that go after these little nematode worms and strangle them. There's a Japanese movie called The Attack of the Mushroom People. 1950, and there we see some real carnivores. You should watch this movie. Yeah. Well, it makes Godzilla look like an Oscar nominee, right? It's pretty B movie-ish, but some of them are carnivores, even if they don't go out and chase down people and eat them. A few of them are actually parasites. They are killing their hosts. For example, down in Oregon, we have a forest down there. A mushroom we call the honey mushroom is eating that forest. It's killed 2,200 acres of trees. And it's all one fungus, right? We believe it's the largest organism on Earth. And it's it makes its living by killing its host, right? So some are parasites. However, relatively few mushrooms are either carnivores or parasites. Most of them fall into one of two other categories. Now, the typical mushroom, when people think mushroom, they think toadstool, and they think of rotters, decomposers. They're out there, they're saprotrophic. And certainly the mushrooms you buy in the grocery store are saprotrophic. They are rotters. The reason they are is because we can grow them on the rotting medium. For example, the button mushroom, I'm sure many of you are aware, part of its life cycle, it grows on horse dung. So that's the rotting medium. We can easily grow it. We don't do very many commercial mushrooms here in North America, but in China, for example, there are at least 25 different mushrooms that are economically significant and sold in the stores. And they're all rotters. They're all decomposers because you can grow them on the decomposing medium, right? So that's what people think about as typical mushrooms. However, I'd like to suggest to you that here in BC, in the fall, that is not the typical mushroom. The typical mushroom is a mycorrhizal mushroom. And it makes its living in a totally different way. Mycorrhizal mushrooms live in a symbiosis with plants. They're not killing the plant. They've got a trade-off going with the plant. I'll explain this in a moment. And these things produce a lot of the wild mushrooms we eat. However, if you're going to pick if you're going to eat mycorrhizal mushrooms, you pretty much have to go out and pick them in the wild. You cannot cultivate them because they live in symbiosis with the host. You'd have to cultivate the host. There is, however, one mycorrhizal mushroom that's cultivated in BC. Do you know what that is? No, but that's a rotter. Yeah, it's a truffle. There are farms here in BC where farmers have planted big groves of oak trees and inoculated them with truffles just to get the truffles. They're not harvesting the oak, right? As far as I know, that's the only cultivated mycorrhizal mushroom. There have been attempts to do some others, but they haven't been very successful. Mycorrhizal mushrooms are at the very cutting edge of research in fungi and mushrooms. Here you see the University of Victoria Library. This is the part of the shelf, this is the shelf on mycology, on mushrooms. And you can actually tell what's hot by the color. Have you ever done this in the library? Years ago, libraries used to actually have money. And when they got a good book, they would rebind the book. So you can see here some rebound books, right? They often bound them in their own drab colors. But then the library budgets got cut. So they started buying hardbacks and taking off the dust jackets. Of course, you know, when you buy a hardback and you take off a dust jacket, it's still pretty drab, but they look a little different, so you get a little more color. So some of these are hardbacks with the, well, let's see, I have to look for them here, with the covers removed. But then, of course, they cut the library budgets again. So all they could afford to buy were paperbacks. And in fact, many books are only published as paperbacks now. And the neat thing about paperbacks, besides the fact they wear out quickly, the bad thing about them, the neat thing about them is you can print the colors in cover for almost no extra cost. So everybody who does paperbacks prints it in color. So if you look on this shelf, look for the color. There it is right there. See that colorful section? That tells me that those are the newer books. And if you go there, what do you suppose that's about? Mycorrhizal mushrooms. Right there. That's the section, right? You can do this with any subject, by the way. If you want to find out what's hot, just look for the color. Let's talk about mycorrhizal mushrooms. There are over 90% of plants, we believe, form mycorrhizal associations with fungi. And as I say, they're mostly tit-for-tat relationships. The plants photosynthesize, they make the carbohydrates, and then they send them down to the roots, and through the roots they pass them on to the fungi. So the fungi get the carbohydrates they need, right? So what are the fungi doing for the plants? They're spreading out through the soil and going out and fetching minerals and nutrients and water and bringing these things back to the plant and giving it to the plant. It's like a little marketplace. They're trading, right? Fungal associations that one study at UBC found can expand a plant's exposure to water and nutrients 60-fold. In fact, take a look at this. This is the cover of the standard text right now on mycorrhizal mushrooms, Smith and Reed. And in this cover, here is a sapling, a seedling of a conifer, which is in a mycorrhizal association. And the roots of the conifer are the yellow parts right here. And that's the only part that's the root. All this other stuff, this white, those are mushrooms. They're spreading out through the soil, absorbing nutrients, and bringing them back to the plant. So you can understand why it would give you a 60-fold access to what's in the soil. So the most common kind of mycorrhizal association is one we call arbuscular mycorrhizas, the AMs. These are in the phylum Glomeromycota, which does not produce mushrooms. The hyphae actually penetrate the roots of the cells. They get together with the roots, and the hyphae go right into the roots. So here's a picture of a plant cell in the root tip, and you can see the hyphae of the fungi have gone right inside the cell. It's a very intimate relationship. These are, AMs are more common in fields, in crop plants, and in the tropics. And there's been some experimentation actually to see if we could introduce these into our crops and make them grow better. And you can now buy packaged arbuscular mycorrhiza spores in the nursery. Do they work? Well, if your plant has not got the associated mycorrhizal partners, they would work. So in other words, if you've got a planter box that's been sterilized, or you just dug up some pavement, this might be a big boost. Otherwise, they're already in the soil, so I don't know. But you can, your mileage may differ. You can give it a try. However, we're not so much interested here in the Pacific Northwest in arbuscular mycorrhizas. We're interested in what's called ectomycorrhizas, EMs, because they're much more important. Ectomycorrhizals, ecto means outside of, they don't penetrate the roots of the cells. They go in between the cells. We know of some 5,000 different ectomycorrhizal fungal species, and 80% of them have visible fruiting bodies. These are our forest mushrooms. So when you go out in October in the forest, you see these mushrooms everywhere. Most of those mushrooms will be the reproductive bodies of ectomycorrhizal mushrooms that are living in partnership with those trees. They're usually in the phylum Basidiomycota, so we're talking about things like boletes, courts, chanterelles, fiberheads, hedgehogs, russulas, tricholomas, amanitas, pine mushrooms. All of these are predominantly mycorrhizal. Okay? And here's how they associate themselves with plants. If you go out to where you have a forest with some Douglas fir, and you dig down in the soil, and you pull up a root, and look at the ends of it, I was told back in biology that you would find root hairs. You're going to be shocked. There are no root hairs. What you've got instead are these larger roots, and they have what look like Q-tips on the end of them. You think, what's that? Well, if you blow it up with a scanning electron microscope, that Q-tip you see is a mass of hyphae from fungi, ectomycorrhizal fungi. They go off in the soil, collect nutrients, and bring them back to this central area, which is where the communication with the plant happens. If we would slice one of those in half, that's that little Q-tip fuzz around it, and you can see they're sending their hyphae in here between the cell walls. Not into the cells, but between the cell walls, and this is where the nutrient exchange happens. That's called a Hartig net. Interestingly, a given ectomycorrhizal fungus out there in the woods may colonize several plants several trees in the same area, even different species of trees. The hyphae of compatible mycorrhizas readily fuse into a single interconnected mesh, and we call this a common mycorrhizal network. It is the wood-wide internet. Okay? The carbon compounds from one plant can be shunted along this wood-wide net into another plant. A lot of work on this has been done right here at UBC by Suzanne Simard and her team. And there are YouTube videos. You can see her team out actually doing this. They will inject carbon compounds that are radioactively tagged into one tree, come back a few hours later with a Geiger counter, and they find it in the soil and in another tree. So one tree is given nutrients to another tree. And sometimes a mother tree will actually be feeding her saplings. They literally are mother trees. You know, we think, oh, it's only animals that nurture their young. Well, apparently, plants can do it too through this hyphal connection. We can actually see this at work in the woods when we go out there in two ways. Have you ever seen these things in the woods? We call them living stumps. They're stumps that have been cut off about that high or have been broken off, and they're still alive. If you take a corer and you put it in the top of that and pull it out, you will see growth rings. Very small, but growth rings. Not only is it alive, that stump is growing. But how is it growing? It's a conifer, so it doesn't resprout. It has no photosynthesis going on at all. Why is it still alive? And the answer is this tree. This tree is photosynthesizing up here. Down come the nutrients into the soil, into the hyphal nets, and they're feeding the stumps around it. Okay? Or what about this? This is something called a mycoheterotroph. These are plants that may have no chlorophyll. Well, a plant with no chlorophyll, I mean, how does it make a living? You know, what's going on here? We used to call these things saprophytes. We now believe there is no such thing as a saprophyte, a plant that lives on decaying matter, right? In fact, what the Indian pipe does, this is the Indian pipe, what it does is it forms an association down at its roots with the common mycorrhizal networks, and it's fed by the trees around it, the bushes. There's a whole bunch of these mycoheterotrophs out there, coral rhizus, all kinds of things. So we can actually visibly see this net at work. Donella Meadows, one of the authors of Limits to Growth and the founder of the Sustainability Institute, when she was told about common mycorrhizal networks, she paused and she said, "What does that mean?" And indeed, I would pose that question to you. What does this mean, these common mycorrhizal networks? It almost seems that the life out there is being guided not by the trees and bushes that you see, but by the mycorrhizal networks in the soil. Those are the ones calling the shots. We really need, when we walk out in our forest, a more mycocentric point of view, a less phytocentric point of view. Sure, there are plants out there. They're fun to look at. They're fun to learn their names. They're there to enjoy and to photograph. They're very visible, but there's really something else going on out there, and it's very important to be aware of this. And forestry management has only begun to scratch the surface of what all this means. Who's really calling the shots out there in our forest? So this is just a brief overview of what's happened in these mycorrhizal studies. Now, the last thing I want to do for you today is the coolest thing, at least from your point of view, and maybe from mine. I want to look at some different kinds of mushrooms. What I showed you earlier was the gilled mushroom, and most of the mushrooms you'll find in the forest are gilled mushrooms, right? Or boletes, which are like gilled mushrooms, except they have pores under the surface. But there's a whole bunch of mushrooms out there that aren't either one of these things, and some of them are mycorrhizal. And so let's just take a quick overview of some of the stranger mushrooms, macrofungi, out there in the woods. Okay? They come in a great many different forms. Okay? And by the way, these are my own pictures. I call them monkey pictures. You know, they say, if you had an infinite number of monkeys and an infinite number of typewriters and an infinite amount of time, they would eventually type all the works of Shakespeare. Yeah, well, I'm not a photographer, but with a digital camera, I can take a lot of pictures, and occasionally one turns out. So here are the different kinds of fungi we find out there. And we don't have the gill mushrooms here. These are the non-gilled mushrooms. So let's take a look at some of these. So first of all, truffles. We have a lot of truffles out there. We train dogs to go out and find these things because they're under the soil. They're hypogeous. True truffles are ascomycetes. False truffles are often basidiomycetes. All of them are mycorrhizals, as far as we know, and as I say, one of them is cultivated. They can be hard to find, hard to identify. Here's one we just found this weekend, Rhizopogon, very common out there. Truffles, they don't have gills. The spore mass is in here, and so you've got to wonder, how do the spores get out to make new fungi? And the answer seems to be that they attract animals to dig them up. And they go through the gut of an animal, many of them. Others may find other ways to spread themselves. It's a puzzle. And then we have a bunch of mushrooms we call polypores. The polypores they have their pores on a non-detachable, usually hard layer underneath. And unlike boletes, where it's a soft layer and you can peel it off with your thumb, the polypores are things like shelf fungi. They're shelves on the trees. We call them conchs. Some are stemmed. Many are resupinate. They lay flat. Some polypores last for several years. A few species can grow to huge sizes. The record seems to be a bracket fungus in Washington that weighed about 300 pounds when it fell. It didn't fall on anybody. So here's a very common polypore. I've seen dozens of these just in the last week. It's called Polyporus batius, or blackleg. I've seen one this big. Most of them are around that big. They're little polypores, and if you turn them over, you'll see this hard pore layer underneath. And if you take a little scope, a little hand lens, and look at it, you'll see the holes that the spores fall out of. And so a lot of our fungi out there are these polypores. Here's one called tiger's eye I found on Salt Spring. They can be quite beautiful. And tiger's eye, interestingly, it'll have multiple stems will come up and the caps will all fuse into one giant cap. Then we have things like turkey tail. It's on that card up there. They're very beautiful. They look like turkey tails. They're supposed to be medicinal. I know a fellow that I haven't seen him for several years now, but he's a true mushroom vagrant. He wanders around from place to place just to be where the mushrooms are sprouting. He starts up in Alaska and works his way down the coast. But when I go out with him, when he sees this, he finds it fresh ones, he pulls it off and chews it. He says it's like chewing gum. I'm like, yeah, okay. I like a little mint in it. And here's one that comes out very early. It's already out now. It's called chicken of the woods, and this is one of the few polypores that's eaten. You can cut off the outer edges of these and fry it up. It tastes like chicken. Okay. Then we have fungi we call jelly fungi. They can be convoluted, club-like, even stemmed, and all of them have a jelly-like consistency, from gooey to semi-hard. Here's one beautiful little one. This is about a centimeter across, called Ascocurrini sarcoides, purple jelly drops. Doesn't that look good? And then we have, of course, the famous witch's butter. You see a lot around Halloween. And the Tremella encephala, the brain tremella, the lighter colored one. These have a jelly-like consistency. And here's one of the most common ones out there, Pseudohydnum gelatinosum. Its technical name is, its common name is tooth jelly fungus. Underneath it has teeth, right? But my friend Andy, in his book, renamed this the spirit gummy bear. And that's kind of what it's like. It's like a gummy bear, only it's the spirit, spirit bear. Yeah, he's crazy too. But it's very common. By the way, this one is edible. I've eaten it. No matter how you cook it, it will not take up any taste, right? So the problem is getting it down your throat, right? It has a mouthfeel that would sink any company that produced it in a commercial product, okay? Then we have the veined fungi, and you'll immediately think of this one. All of our chanterelles, they don't have gills, they have veins underneath. Gill is the same size all the way down. A vein is more triangular and sometimes very, very shallow. And these are out now. I just saw a whole bunch of them this last weekend. And then there's this one, Gomphus floccosus, the woolly scale chanterelle, also called the false chanterelle. Many people mistakenly pick this one thinking it's chanterelles. I know I was out in the woods around Duncan a few years ago with a science group, and I saw this guy with a bucket coming through picking. And I said, oh, what are you picking? And he said, oh, chanterelles. And I went over and looked in his bucket. It was all this stuff, right? And this is, in fact, for some people, poisonous, right? Apparently not for him. Other people eat this, so I don't know, right? Then we have the corals and clubs. The corals and clubs differ in the sense that club fungi are generally unbranched or lightly branched, where corals are multiply branched. They look like corals under the sea. In this case, the spore-bearing basidia line the outer surfaces of many of the corals and clubs. The ones with more obvious heads are actually ascomycetes. They come into vast ranges of sizes and colors. The one you most often see out there is Ramaria, and Ramaria are ectomycorrhizos. Here's a club fungus I like a lot. It's truncate club coral. I find this a lot in Francis King, and if you pick this one and just tear off a little of the outside and put it in your mouth, it's like saccharin. It's that sweet. It's just a burst of sweetness. Fairy fingers. This one we once thought was rare. I'm beginning to doubt it because I'm seeing it everywhere. But a beautiful little thing. It grows in damp areas, Mitrula elegans, earth tongues. I've actually seen this growing underwater. And some of the club fungi are cordyceps. Now, most cordyceps, they grow on the dead bodies of caterpillars and animals. And they kill the caterpillars, right? However, a very common one we call the truffle eater because it grows on truffles, right? Did you know that? You're out there looking for the truffles, and you see this club fungus, and yeah, there's a truffle under there, but it's pretty much gone, right? This thing is feeding on it. Cordyceps are quite important in Asia because they are used medicinally, and so there's a great business going out and finding dead caterpillars that are sprouting mushrooms. Here's a beautiful Ramaria, Stuntzii. There's some shockingly beautiful ones out there. And this is one, I just found one of these the other day. This is a cauliflower mushroom. We put this in the coral group. This guy is smiling because our Southern Vancouver Island Mycological Society, we go up to Cowichan every year, and he always comes along. And as soon as we go out in the woods, he disappears. And he comes back a half an hour later with a big smile on his face and holding one of these cauliflower. They're delicious, right? And they come up in the same place every year, and he won't tell anybody where it is, right? And I never see him the rest of the year. He only comes to this thing, and so anyway, I took a picture of him. Then we have cup fungi, really interesting. Little, they're small. They're ascomycetes, and they belong to many different families. Here's one called starving man's licorice. It really does look like licorice. It is not, however, edible, and it does not taste like licorice. It just looks like licorice. And then we have this pig's ears, another cup fungi. You have to turn it over to see that it's a cup. And then we have the blue stain. A lot of carpenters love to find wood with this blue stain in it. It has character, right? And every once in a while, the blue stain will put out a reproductive body, and they're cups. They're little blue cups. And you see these every few years popping out. Some cup fungi are actually enclosed, like this one, the crown fungus. It all forms a little ball out there. And some animals, some slugs, live inside. And then we have crust fungi. Crust fungi, they're like small polypores, but without a pore layer. They have wrinkled or a warty pore-bearing surface. Many form parchment-like sheets on logs, and they're almost all saprotrophs. For example, here's one called coral spot. Beautiful little coral-colored spots. And then this one is actually, this is a lobster mushroom. In my opinion, one of the most delicious mushrooms in the world, but it's actually two mushrooms. It's a Russula, and the Russula has been infected by one of these crustal fungi called Hypomyces lactuiflorum, and it eventually replaces every cell of the other mushroom. It keeps its shape, but gives it a completely different taste and texture, and it is delicious. And you can find these in the woods. We have Hymenocata. You see them growing on branches. Then we have, of course, the ones that look like brains, the morels. They usually come up in the spring. The morels do at least. And they often appear in areas where there's been a burn. Here's one, Geometra, which is the false morel, and some people accidentally pick this. It is actually edible, but the problem is it contains hydrothiazine, which is jet fuel. And you got to get rid of that. In fact, people have poisoned themselves by cooking this in a closed room. And the jet fuel vaporizes, and people flop, right? And wake up hours later, right? What happened? But if you can get rid of the jet fuel, they tell me it's actually edible. I'm not that anxious to try it. And then these little elfin saddles, which they now say are edible. They used to say they weren't. I've never tried one. Beautiful little things. We can imagine an elf riding on a saddle. And then we have the puffballs. Their fruiting body is a large spore case, but there's usually no stem. And they have an inner and outer layer on their skin, and the spore mass is inside of them. Initially, it's white, and many of the puffballs, when the spores are white, they're edible. But then they turn greenish, looks like something out of a horror film, and then they turn black and dry out, and at that moment they're ready to puff. And the way they puff is when raindrops hit them, a little hole forms in the top, and the black spores will come out in a little puff. This one, for example, is a dark puffball, and it's Lycoperdon. Lycoperdon means wolf fart in Greek. Yeah, well, you get the idea. The rain hits it, right? So, some of these are beautiful. I saw a bunch of these just yesterday. Geastrums. They're puffballs, but they have this outer skin that separates, and it forms a star around them. It kind of looks like an eyeball, doesn't it? You can see these. And then we have the bird's nest fungi. They're very small, and this is only maybe a centimeter across. And they have these eggs in them. Now, those eggs are not spores. Those are spore cases. There's hundreds of spores in those things, right? But these are splash cups. So when the rain hits this thing, it throws out the spore cases. They're slimy. They hit plants and stick, and then they dry out and drop their spores. But for the brief period of time that they have the egg cases in them, they really do look like bird's nests, don't they? With eggs in them. And then, of course, we have the toothed fungi. They have a cap. Many of them have a cap and a stipe, just like gill fungi. But underneath, you'll find these little spines hanging down. Many are tough, but some of them are actually good edibles. Here's a beautiful one. Herasium abiatus, bear's head, wonderful tasting one. It will come up in the same place every year if you know where one is. This one is at Royal Roads. I haven't checked this one yet. Doesn't it look like icicles falling down? Yeah. And here's a little one that is one of the most delicious mushrooms in the world. It's a hedgehog. It's a mycorrhizal, and it has these spines underneath. And it's a safe mushroom. Those of us who take others out on walks and show them fungi, we differentiate between safe mushrooms and safe edibles and unsafe edibles. Safe edibles are the ones that beginners can learn to identify, and there's no poisonous lookalikes, right? Like a lobster mushroom. right? And this is a safe edible. There's nothing like it. If it has spines in that color, right? And last, let's just take a look at boletes. A lot of boletes out there, some of the best mushrooms in the world. They have pores, and there's a tube above the pores, and the spore falls down the tube. It's held in a fleshy layer under the cap, and you can remove it with your thumb. Around here, the most common members of the bolete family are the Pseuilus, Boletus, and China mushrooms. Say many are good edibles, but flies love these things. So you got to find the mushroom before the flies do. They lay their eggs, and pretty soon there's maggot holes all through these things. One of them I really like is Boletus mirabilis, lemony taste and smell. Very common around here. Usually comes out of some rotting wood. This is one of those funny ones that we think is both we know it's mycorrhizal, all boletes are, but we think it's also partly saprotrophic. Or at least that's where it's gathering the nutrients for it to share with its partner. And then, of course, the king bolete. This is the one you buy in the stores, and it's called a porcini. Wonderful nutty flavor. It's a safe edible. Nothing looks quite like it, right? And it can be huge. I mean, this thing in the bottom can get this big, and the top can get this big. But usually by that time, they're maggoty, right? And right after I finish, Daryl's going to show me his favorite bolete spots, right? And anyway, when you know your spot, you go back to it year by year. It's a wonderful safe. However, 5 years ago, 11 people died in Italy because of this mushroom. They didn't eat it. They went out at night trying to steal from other people's spots with flashlights, and they fell off cliffs. Right? So there are many different ways mushrooms can kill you. They don't all poison you. Okay, so that's a brief review of some mushrooms that we have around here. Do we have time for any questions? Yeah, okay.